Southwestern Germany, 2009. A construction crew sets a blasting charge in a limestone quarry. When the dust settles, there is a fossil in the rubble shattered into a jigsaw of odd-sized blocks. Somebody collects it, it goes into a small regional museum, and there it sits. 16 years later, a team finally finishes reading it. What comes off that slab is not bone. It is skin from the flipper of a 10-meter sea dragon that died 183 million years ago, and it carries 4 features that have never been found together on any swimming animal, living or extinct. Getting from that pile of rubble to that conclusion took 24 scientists, a Nature paper, and a particle accelerator in Switzerland. But the story does not start in a German quarry. It starts on a cliff in Dorset, 214 years ago. In 1811, on the crumbling blue lias of Lyme Regis on the south coast of England, Joseph Anning found a skull. His sister Mary Anning worked out the rest of the skeleton over the following year, digging it free while winter storms kept exposing new sections of cliff. That specimen became the first Ichthyosaur ever examined by science. It is the reason the word Ichthyosaur exists at all, and it belongs to the same genus as the flipper in the German rubble. The genus is Temnodontosaurus, a parvipelvian ichthyosaur, one of the derived and fully fish-shaped members of the lineage. The group had spent something like 60 million years remodeling itself away from a 4-legged land ancestor into something that looks, at a glance, like a shark or a dolphin. Limbs became fins. The tail acquired a crescent fluke. The body became a streamlined tube built for open water. Susanna Guttara and colleagues at the University of Bristol modeled that transition in Proceedings of the Royal Society B in 2019, running digital reconstructions through simulated flow to measure how the drag cost of swimming changed as the body plan evolved. Skeletally mature individuals occasionally passed 10 meters in total length, roughly the length of a large bull orca, a figure documented by the paleontologist Christopher McGowan in his 1996 review of Early Jurassic giants in the Canadian Journal of Earth Sciences, and reinforced in 2023 when Nigel Larkin and colleagues published the excavation of the specimen known as the Rutland sea dragon from the English Midlands, the largest ichthyosaur skeleton yet recovered in Britain. It was the apex predator of its sea. Work by Rebecca Bennion, Aaron Maxwell, Olivier Lambert, and Valentin Fisher, published in the Journal of Anatomy in 2024, examined the skull and teeth across the genus and placed it at the top of the early Jurassic food web. Gut contents back that up. In 2025, Giovanni Serafini and colleagues published a study in Papers in Paleontology on Temnodontosaurus bromelites, the fossilized digestive residues, and recovered the remains of squid relatives and of other ichthyosaurs. And then there are the eyes. Ryosuke Motani, Bruce Rothschild, and William Wall reported in Nature in 1999 that the sclerotic rings of Temnodontosaurus indicate eyeballs exceeding 25 centimeters across, which is wider than a dinner plate. As far as the published record goes, those are the largest eyes measured for any vertebrate, animals alive today included, rivaled only by the giant and colossal squid. An eye that size is a light-gathering instrument. It is what an animal builds when the water it hunts in is dim, whether that means depth or darkness, so the vision half of the picture has been on the table since 1999. The questions this genus usually gets asked are about size and about bite force. Both are reasonable questions, and both are hard to answer well. Fragmentary giant bones exist, and scaling a whole animal from an isolated vertebra or shoulder blade produces numbers that vary a great deal, depending on who does the scaling. Bite force figures for the genus circulate widely, but they are largely scaled estimates rather than the output of a dedicated study. The peer-reviewed craniodental work by Bennion and colleagues describes jaw and tooth shape across the species, and what that implies about diet, which is a more careful claim than a single headline number. What was missing from all of it was everything soft. That gap has a reason, and the reason is preservation. Ichthyosaur body outlines do exist. The black shales of the Posidonia formed under an oxygen-starved seafloor during the early Toarcian, the interval Hans Joachim Röhl and colleagues characterized in 2001 as an oxygen-depleted ecosystem controlled by sea level and climate. The paleontologist David Martill described the mechanism in 1993 as soupy substrates, seafloor mud soft enough for a carcass to sink partway in and be sealed off from scavengers. The setting behind that is a global event. Around 183 million years ago, the oceans went through the Toarcian oceanic anoxic event, a warming-driven collapse in seafloor oxygen that killed off marine invertebrates across large parts of the world. What it produced for paleontology is an accident of chemistry. Water with no oxygen in it has no burrowing animals in it either, and nothing to pull a carcass apart, so what falls to the bottom stays articulated and stays chemically intact for far longer than it has any right to. Aaron Maxwell and Peggy Vincent tracked what the event did to the ichthyosaurs themselves in paleobiology in 2015. They found no species-level extinctions among ichthyosaurs at the peak of the invertebrate crisis, but the large-bodied animals disappear from the German basin through the worst of it and become more common immediately afterwards. Whatever was happening to the ocean, the big predators moved out and then came back. The flipper in question sits in a specific layer of that sequence, a unit German geologists call the Unterer Stein, and dates to roughly 183 to 181 million years ago. It is exactly the right rock for soft tissue. The problem is that the outlines which survive belong overwhelmingly to smaller animals. Stenopterygius, roughly the size of a dolphin. In 2018, Johan Lindgren of Lund University led a team that pulled remarkable detail out of one of those Stenopterygius specimens, publishing in Nature evidence for scaleless skin, insulating blubber, and pigment cells arranged in a countershaded pattern. Mats Eriksson and colleagues reviewed the whole Ichthyosaur soft tissue record in Earth Science Reviews in 2022, and the pattern held. Small and medium-bodied animals were represented. The megapredators at the top of the system were not, which is why the German flipper matters, and why it took so long to read properly. The collector who recovered the slab in the first place was Georg Göltz, and the specimen went into the Paläontologisches Museum Nierstein in Nierstein, Germany. Initial preparation began when it was found. Then, in 2021, an investigative computed tomography scan showed that more bone was still buried inside the matrix. Preparators went back in with chisels, dental picks, and a microscribe. Dean Lomax, an ichthyosaur specialist and research fellow at the University of Bristol, worked on the fossil for roughly 6 years. The final study, led by Johan Lindgren at Lund University with 23 co-authors, was published in Nature in July of 2025. What they had was a forelimb about a meter long, longer than a human arm from shoulder to fingertip, carrying 4 digits plus an extra accessory digit along the rear margin, with the radio, 1 carpal, 1 metacarpal, and the phalanges still in near-perfect articulation. The proportions and the notching along the leading edge identified it as Temnodontosaurus trigonodon. The bones were only the frame. Coating them and extending well past them was a dark film of mineralized soft tissue that recorded the actual outline of the fin in life. Establishing that the film really was skin, and not just a stain, took a stack of techniques that has become standard in this corner of the field and is worth naming. The fossil was photographed under polarized light, and again under ultraviolet, which makes phosphate-rich tissue fluoresce against the surrounding rock and pulls the outline out of a slab where the eye sees almost nothing. Sections were ground down to optical translucency and read under a microscope. Field emission scanning electron microscopy resolved the fine structure. Time of flight secondary ion mass spectrometry, run at 3 separate instruments in Sweden, identified the chemistry of the residue by firing an ion beam at the surface and weighing what came off. Infrared microspectroscopy was performed partly at the Soleil synchrotron in France. What came back was a layered structure, outermost, a phosphatized layer with brown specks scattered through it that resolved under electron microscopy into cell-shaped casings, some with branching arms, each packed with dense granules. The chemistry matched eumelanin. Those are melanophores, pigment cells, and the granules inside them are melanosomes. Below that sits a compressed band of tightly packed melanosomes representing the boundary between the outer and inner skin layers, flattened by burial. Below that again, bundles of structural fibers running in an ordered mesh. So this is skin in section from a 10-meter marine reptile that died in the early Jurassic, and that outline was not the shape anyone had drawn. Historical slab-mounted skeletons of this species show the digits converging towards the tip, giving a paddle. In this specimen, the digits stay nearly parallel down their full length, and the soft tissue continues beyond the last bone into a fleshy point with no skeletal support inside it at all. The result is long, narrow, and high aspect ratio. It is a wing rather than a paddle, and it is the first direct evidence of that shape in a large ichthyosaur. Here is the one question worth dropping in the comments. Before this fossil, every reconstruction of this animal was drawn from bone alone. What else in the standard picture of Mesozoic marine reptiles is an outline nobody has actually seen? By the time the microscopy work was done, the fin had produced 3 more features, and each one is stranger than the last. The first is on the surface. Running across the entire blade of the fin, from leading edge to trailing edge, are evenly spaced stripes set roughly 2 millimeters apart, about the thickness of 2 stacked coins. They never intersect, some of them fork. The obvious objection is decay, because rotting skin loses tension and wrinkles, and the authors address it directly. The spacing is too uniform, the distribution too even, the underlying fiber bundles too repetitive, and nearly identical lines appear on a forelimb of the closely related Ichthyosaurus held at the Staatliches Museum für Naturkunde in Stuttgart. The evidence indicates the stripes were on the living animal. The second feature is along the back edge. About halfway out along the span, the trailing margin stops being smooth and becomes a row of pronounced sinusoidal serrations, like the teeth of a bread knife rendered in soft tissue. The third feature is what holds those serrations up, and it is the part that required a new word. Embedded in the skin along the trailing edge is a series of rod-shaped mineralized bodies. Near the base of the fin they are slender and spike-like, about 4 to 6 millimeters long, roughly the length of a grain of rice. Further out they enlarge to 9 to 13 millimeters and take a pointed conical shape, about the length of a staple. At the very tip they flatten into small oval plates, 2 to 4 millimeters across, smaller than a lentil. Structures in the skin are not unusual for a tetrapod. Crocodilians have them. They are called osteoderms, and they are made of bone. These were not. Under microcomputed tomography, synchrotron imaging at the Swiss light source, and electron microscopy, each element resolved into densely packed globules containing chambers and paired cell-like bodies, matching the size and arrangement of chondrocytes, which are cartilage cells. Petrographic sections showed concentric growth banding consistent with successive waves of calcification. Chemical demineralization released cellular material and remnants of extracellular matrix. Bone in the skin is an osteoderm. This was calcified cartilage in the skin, and osteoderm development does not pass through a cartilage stage, so the term did not apply. The authors proposed a new one, built from the Greek for cartilage and the Greek for skin, chondroderm. One short aside and then straight back to it. This channel is new, and an episode like this takes a long time to research properly. If careful work on a single fossil is the kind of thing you want more of, then a like and a subscribe is the only favor I will ask for, and it helps more than it looks like it should. Thank you. 4 features then on one fin. A long, narrow wing planform, a fleshy tip with no bone in it, chordwise stripes across the whole blade, a serrated trailing edge stiffened by cartilage rods. No aquatic vertebrate, extinct or living, has ever been recorded with that combination. The next question was what the combination was for, and the answer came from fluid dynamics rather than paleontology. Every one of those 4 features maps onto a known noise suppression device. The flexible, unsupported tip behaves like a winglet, bending under load, reorganizing the vortices that spin off a fin tip, and reducing the drag and the sound those vortices generate. Surface riblets and troughs of the kind the stripes describe are used to interfere with turbulent noise sources. Serrated trailing edges are the classic case. They are the reason a barn owl can fly at a mouse without the mouse hearing it, and that mechanism has been studied in detail by Justin Jaworski and Nigel Peake, whose review of silent owl flight ran in the Annual Review of Fluid Mechanics in 2020. There is a detail here that is difficult to write off as coincidence. In the fossil, the serrations are most pronounced at around 75% of the way out along the span. That is the same position they peak on the trailing edge of an owl wing, and the same position the crenulations peak on the tail fluke of a humpback whale. So the team tested it. Robert Zoltan Saaz and Johan Revstedt, both engineers at Lund University, ran computational fluid dynamics simulations on a virtual section of the fin, taking chord length, serration size, and spacing directly from measurements at that 75% station. Flow speed was fixed at 1.5 meters per second, near the estimated efficient cruising speed for an ichthyosaur of this body plan. Angle of attack was tested at 0, 5, 10, 15, and 20 degrees. Sound was estimated using the curl acoustic analogy, with a virtual microphone placed 50 meters upstream, which is where prey would be. Adding the serrations reduced emitted noise by roughly 1 to 3 decibels across low frequencies below 200 Hz, and did so with negligible cost to lift and drag. Adding the surface ridges or troughs on top of that produced a further reduction of 5 to 10 decibels, most pronounced below about 70 Hz. 10 decibels is not a small number. In human hearing, a drop of that size is roughly the difference between a sound and half a sound. Low frequency, quiet, no penalty to swimming performance. That is the signature of a system built for stealth rather than for speed. And the frequency band is the part that closes the argument. A noise reduction is only useful if it falls inside the hearing range of whatever the animal is trying to sneak up on. Temnodontosaurus ate squid relatives, confirmed by the gut contents Serafini and colleagues published. Cephalopods are sensitive to exactly this part of the spectrum. Andrew Packard and colleagues demonstrated low-frequency hearing in cephalopods in 1990, and Aaron Mooney and colleagues showed in 2010 that the long-finned squid responds to low-frequency particle motion. The picture the authors assemble is a large-bodied predator gliding on high lift fins in dim water, keeping tail movement to a minimum because the wings are doing the work, and suppressing the low-frequency sound of its own passage in the band its prey can detect. A squid is small and fast and can outmaneuver something 10 meters long, but only if the squid knows it is there. Remove the warning and the size advantage becomes decisive. There is a second consequence the authors raise, and it runs in the opposite direction. A quieter animal is also a harder animal to find. Temnodontosaurus was the apex predator of its own sea, but a subadult was not, and the gut contents show that members of this genus ate other ichthyosaurs. Suppressing a hydrodynamic signature works as concealment from something hunting upward, as well as concealment from something being hunted. There is a third, subtler one. An animal that generates less turbulence around itself is an animal that is interfering less with its own sensing. Ichthyosaurs are not thought to have had sophisticated directional hearing, but they very likely detected low-frequency sound in the water. And there is a more speculative suggestion, based on structures resembling scale organs in living reptiles, that they may have had skin receptors reading water movement directly. A predator that swims loudly is a predator swimming inside its own noise. Put those together and what the paper proposes is an arms race. Prey animals in the early Jurassic getting better at detecting an approach, and at least one lineage of large ichthyosaurs answering with a package of low-light vision and acoustic suppression at the same time. Neither half of that is much use alone. Eyes that work in the dark do not help if the animal announces itself. Silence does not help if it cannot see. The rigorous version of this comes with limits, and the paper states them. The individual was a subadult, not a fully grown adult. Soft tissue survives on one side of the flipper only, the side pressed into the seafloor mud. The extent of the trailing edge had to be estimated by interpolation. The flattening of the fossil makes it impossible to tell whether the surface ornamentation stood proud of the skin or was sunk into it, so both were modeled. The fluid dynamics work is simulation, not a physical experiment, and the authors flag its inherent limitations. Chondroderms are so far known from this genus and nowhere else, though the curator Mark Jones at the Natural History Museum in London has noted that smaller specimens in that collection show comparable structures and are worth examining. Lindgren has said the next step is more simulation, and ideally a physical replica tested in water. There is one further wrinkle, which is that the argument for the eyes and the argument for the fin depend on each other. The stealth interpretation rests on the animal having hunted in low light, which rests on the eye evidence from 1999. And researchers still disagree over whether eyes that large evolved for fine visual acuity or for low-resolution sensitivity in darkness. The consensus is only that they worked at low light levels, either at night or at depth. Within that consensus, the flipper makes sense. Outside it, the case would need rebuilding. What is not in dispute is the anatomy itself, and the anatomy is now doing work outside paleontology. Shipping traffic, sonar and offshore construction have raised ambient ocean noise to the point where it interferes with the animals living in it. Serrated trailing edges are already being trialed on propellers and hydrofoils to quieten them. The Lindgren team's closing observation is that the same solution was in the water 183 million years ago, worked out by a reptile. 214 years ago, a brother and sister working the cliffs at Lyme Regis pulled this animal out of the rock and handed science its first ichthyosaur. The skeleton has been in museums ever since. What was missing was the millimeter of skin around it, and that arrived in a load of blasted limestone, split down the middle, waiting 16 years for someone to finish reading it. Till the next find, this is Bone Atlas.